Well, thank you so much, and thank you for the honor. And some of my favorite people in the world are in this room right now, for sure. For sure, Bob and Sangeeta are among the top. Um, so, one of the most alarming findings in climate science, particularly atmospheric climate science, over the last 15 years, is related to the fact that as temperature has risen over the surface of the planet for the past century, relative humidity has actually not changed. And the reason essentially is that the Earth is not a ball of water. But the consequence is that the thirstiness of air, which is defined as the difference between the saturation pressure at any given temperature and the vapor pressure, is going up exponentially with rising temperature. And that means that there's major threats to ecosystems on the planet. This didn't become clear until around 2000, where you can sort of see the dip, particularly over the drying surface of the planet of relative humidity. And particularly, people began to monitor vapor pressure deficit. In the early 2000s, the first papers came out in the early 2010s, and there were a lot of groups trying to figure out, well, are we seeing a real phenomenon here? And the reason why that's so important is illustrated in this classic vapor pressure versus temperature curve. So what you see is the red line or the blue line is classic saturation pressure curve. It goes up almost exponentially as you get near to the boiling point of water. And so at 70% relative humidity, which is the average relative humidity on the planet, the vapor pressure deficit at 20 C is about 700 pascals. Now that's a healthy vapor pressure deficit, and ecosystems have evolved with that vapor pressure deficit. However, when you go to 40 C, exact same relative humidity, the vapor pressure deficit is 3 times greater. So it's 3 times thirstier air. And for reasons that we'll describe in this talk, that's lethal to many ecosystems on the planet. What's making matters worse, and we feel them now, and it's showing up in reports of asthma exacerbations and influenza incidents with heat waves, is that the incidence and the duration of heat waves is increasing in also an accelerating way with rising temperatures. A recent paper came out in Nature indicating the burden of heat waves that is projected to occur over the next 50 or 60 years. And they concluded that by the end of the century, whatever your hypothesis is about the rising temperatures, crop failure rates would be around 40 to 50% per year. And we're going to come back to that number a little bit later. Now, among ecosystems on the planet that are suffering and that you hear most about are plants, because we know most about why they're suffering. And the reason they're suffering is something called transpiration. Transpiration is an amazing phenomenon. Most of the water that leaves the surface or the soil of the earth leaves by transpiration. And it occurs in the following way. When water evaporates over hydrogel, macromolecules of the hydrogel get pulled to the evaporating surface, and they create the osmotic pressure gradient, and it has 2 effects. It creates a suction, pulling water from beneath. So in the case of a plant, the suction pressures can be thousands of atmospheres, so pulling water from the distant soil, which is great. It also puts a pressure on cells within the extracellular matrix, which in normal conditions is good. It keeps the plant growing. However, when the vapor pressure deficit becomes too high, you get inflammation, the stomata close, the leaf wilts, and it eventually dies. Now, we know a lot about how plants respond, and particularly, and by the way, this phenomenon, which is generally useful, is used in lots of industrial applications like water filtration. So this is an illustration of plant existence in the window of existence. So as temperature goes up, vapor pressure deficit goes up. And at about 30-something C, at 60% relative humidity, plants begin to wilt. Some wilt at a higher vapor pressure deficit, but generally that's about what happens. And once you get to about 3 kilopascals, the plants die. Now, if you keep the temperature fixed, 25 C, and just go up in altitude, pressure goes down, vapor pressure deficit goes up. And so the reason why you have a tree line is vapor pressure deficit. And so it's often called the vapor pressure deficit line. And so we'll come back to that too. Now, it's not only plants that are suffering, rodents are too. And while it hasn't been really understood why, generally it's understood. So small rodents, as any of you who do mice experiments know, are very sensitive to dry air. So they have very little humidification capacity in their airways, and while they breathe through the nose, they do so with such a small amount of humidification capacity. It's like us breathing through our mouths. And so the consequence is that mice died at room temperature below about 30% relative humidity, and therefore at cold temperatures they hibernate. And the reason is when you breathe in cold air, even though there's like no vapor pressure deficit, when you breathe it in, you heat up the air. There's no water, and so it's like really hot, dry air. And so they hibernate, and they tend to just die at high temperatures. These two rodents have weird hibernation patterns, and they are getting screwed up by what's happening with climate change. Our work, all of this is a preface to say that our work over the last 5 years has shown that mucosa exposed to the atmosphere are drying out like the topsoil is drying out. And they're experiencing stresses that are exactly the same stresses that plants are experiencing owing to transpiration. And it's a particularly big problem for humans. It connects with lots of phenomena that you're probably aware of, that in cold temperatures and hot temperatures and dry environments, even at high altitudes. So the 40% of Himalayan hikers develop chronic cough. And so all of that is related to vapor pressure deficit. But the reason why it's such a problem is that independently of climate change, we have been moving to indoor living over the last 100 and something years. Asthma, when it first began to be diagnosed in the 19th century, was considered a rich man's disease. And basically, people were living in homes, and of course, there's dirt in the air and all of that, but it's also very dry air. We're also moving to mouth breathing, which is a major problem. So we lose the humidification capacity of our noses, and then there's this progressive shift to urban living. So there's multiple factors that make this an urgent problem to both understand and to solve. So I'm going to spend 20 minutes going over the basic biophysics of mucus transpiration inflammatory cascade. And then we'll talk about how we use that information to treat respiratory diseases that are provoked by dehydration, in particular we'll focus on chronic cough. And then I'll end with some thoughts about what we can do in the future. So what my eureka was, I'm a fluid mechanicist by background prior to meeting Bob, and he de-learned me of everything that I needed to be de-learned of. But I did remember some things. And so when this paper came out in the Journal of Fluid Mechanics several years ago, I was really struck. And so they did these, so the experts in fundamentally understanding transpiration are mostly at the University of Cambridge in physics and applied math. And they did this really nice paper where they took hydrogel beads and they wicked them to tubes of water, and they blew air over the beads. And when the air was dry, the beads shrunk. But they not only shrunk, the head of water that was being pulled by this evaporation got bigger. So you're basically, it's like you're creating a bigger column of water as you're getting more evaporation. So there's like the pressure is going up. And when they is humid, they expanded again, and the pressure went down. And so it was this direct measure of this transpiration problem. And they described it really well in the context of hydrodynamics and the creation of these osmotic pressure gradients. So I immediately thought that, well, we should take a look at whether mucus and airways might be doing the same thing, but with eyes, imagine, have similar phenomena. We're going to focus on airways. And I'm just going to summarize the basic conclusion. I'll go through some more details here, but basically this is what we've learned. So I don't know how many of you know something about airway hydration. Actually, are there anybody here? Not really. Okay, good. So it is often said, and it's true, that airways stay hydrated by ion exchange with the epithelial membrane, and that's true on a breath-to-breath basis, but over many breaths, it's not true. So airways stay isotonic where it relates to salt. So the only thing that's pulling water into the airways, globular protein and tethered mucin, and they create about a 500 Pascal osmotic pressure. And you can show that that's just enough osmotic pressure to get the water that supplies evaporation on nasal breathing. So your airways have evolved to basically pumping water into airways so that you can evaporate it on nasal breathing. and keep your lungs humidified and survive. So this is like a really fundamental thing. Now, what happens is if you now begin to breathe through your mouth and it's not really humid air, the environmental conditions pretty much get right to your larynx, and the evaporation rate is too high, and so the mucus collapses. There's not enough water. But what happens is that as it collapses, the cilia start to, and the tethered mucin get tangled, osmotic pressure goes up, and it pulls water. And so you keep the airways hydrated, and so that's a really functional response of the airways. But the negative is that that compression of the cilia is inflammatory. In other words, it creates pressure. And decades of work by Jeff Drazen and others at Harvard School of Public Health elsewhere have shown that, well, if you compress airway epithelial cells, they eventually get very inflamed. And that's, in fact, what we've found to be true. We showed in our paper this spring that couple big sources of inflammatory pressure. One is just mucin getting, or the globular protein getting more concentrated. And then if you're like running, it turns out this aggregation of mucin near the surface creates like really massive stresses. So most Olympic athletes cough, and they mostly go to bed coughing. And so there's a major problem with athletes, as you can imagine. But we're going to focus on normal tidal breathing in this presentation. So we did experiments in this paper at UNC, which is basically Vic Boucher's lab at UNC has studied airway dehydration from a cystic fibrosis perspective for many decades. And so what's going on here, I see somebody nodding. So what's going on here with normal breathing is the airways can dehydrate, not just because they can't produce like water flux from the epithelium. Remember, there's just too much water leaving from the surface. And so what we showed is we took airway lining interface cultures of human bronchial epithelial cells and exposed them to what classical 95%, 60% relative humidity, or 30%. And you can just show by basic mass conservation that you're going to get thinning of the mucus that happens, but you also get the compression of the periciliar layer, and you can predict quite accurately the reduction in height of the airway stiffness liquid. The disconnect at the high compression we show in our current paper relates to the compression that's added by the cilia themselves. So in principle, you can predict quite accurately. So you get this really interesting, the condition is you're in your home at night, you're breathing through your mouth, and you spend a night breathing through your mouth. And so what happens in those conditions is you're getting compression of your airway epithelial cells. And we show that, as in the Drazen and colleague studies, you get the rapid secretion of inflammatory cytokines, and in this case, INF-alpha, IL-6, IL-33, all implicated in the pathogenesis of chronic respiratory disease, and particularly asthma. So we wanted to know, well, would this happen in an in vivo situation? And so we took mice, and mice, given their vulnerability, they're easy to study. We looked at both mice who had dehydrated airways. In one of my slides, I showed that when your airways are dehydrated, they're especially sensitive to these stresses. And we put them in two groups. One group was just 3 weeks humid, warm air, and one group had twice a day, 15 minutes of dry air. So just 15 minutes of dry air twice a day for 3 weeks. And what we found was massive infiltration of the inflammatory cells, eosinophils, neutrophils, lymphocytes, and consolidation after 3 weeks, particularly in the dehydrated airways. And so all of this is pointing to an inflammatory cascade, which has been studied in the airways from the context of asthma by the Harvard School of Public Health folks. But what we're showing here is that in the larynx and the trachea that are the front line here for the environment, this cascade leads to release of activators of cough receptors on neurons in the vicinity of the larynx and the trachea, and provoke cough. And so cough and cough hypersensitivity are one of the kind of the canary in the mine of dry airways. And of course, what's mediating this is the compression of mechanosensitive ion channels like TRPV4 and piezo1, but also CFTR and so forth, all of which I will talk about in just a little bit. So the issue of chronic cough is a big one. So up until the 1970s, you really didn't see anything in the literature about chronic cough. Same thing about dry eye. The definition of chronic cough didn't exist until the early 2000s, and it's defined as a cough that persists more than 8 weeks. Less than that, it's acute cough. And there's a big percentage of that cough which is unexplained, and it's called refractory or unexplained cough, about 40% of cough. Many of you may have encountered chronic cough in the COVID-19 era, but it's about 10% of the adult population, and it's without a safe and effective treatment. The morbidity is similar to that of COPD, so there's quite an interest in the FDA, payers, and physicians to find a solution. Now, it's ultimately a pain mediation treatment, and so there's a lot of side effects that are associated with treatments that are going through the clinic. And so we thought we might be able to address this by addressing inflammation at the top of the cascade. All right, so I'm going to now talk about what do you do? Now it's known, and before I kind of confuse you with this slide, I'm going to just go back a slide and just talk for a couple seconds. So we know from cystic fibrosis that we can hydrate airways by inhaling hypertonic saline. And it's not used too much anymore for reasons I can describe, but generally it's been used a lot for cystic fibrosis. And it happens that when you hydrate the air, we breathe in hypertonic saline, pulls salt, pulls water into the airways. It gets the mucus off the cilia, you get movement, and it lasts pretty long. It lasts very less than 30 minutes for normal people, but like for several hours for cystic fibrotic patients. And a nice study came out by Brian Button, who collaborates with me and us, and several years ago in vitro. It was really amazing, where they took airway lining interface cultures of cystic fibrotic cells, hypertonic saline, and then normal cells. Normal cells, hypertonic saline, you get this increase in volume of surface liquid of, say, X. You put the same amount of water on the cystic fibrotic airways, and you get like 5X. You get way more water. Well, where is that coming from? So cystic fibrosis is characterized by a very high concentration of mucin, has a lot of globular protein. And so what we have found, and I'll show you a little bit later, that what's happening is you're lifting this mucus off the cilia. You're liberating quite a bit of globular protein, which is sequestered in the mucin interactome. And that globular protein is acting like osmotic pumps, it's pulling water out. And so we said, well, could we, all of which is to say that the goal here was, okay, could we, in normal airways, you don't have all that globular protein, but you have quite a bit. Could we somehow engineer the mucus in normal airways by a hydrating salt solution to release a lot of globular protein for long enough to get a lot more hydration, and enough hydration to be therapeutic? So the question then became, well, what is enough? How long? How long do you need to hydrate airways for that to be therapeutic? So we guessed that there would be an effect if you exposed airways to dry air for some period of time, say 8 hours, the heat wave, 8 hours, and you then look at the expression of mechanosensitive ion channels associated with cough and cough hypersensitivity. Presumably, there would be some period of time during which the airways defended themselves, and then some period of time after which they couldn't defend themselves. And so we looked at 23 mechanosensitive ion channels, and these are just a subset of them. But generally, we found that after about a few hours, if you look in detail here, you can argue that the airways are defending themselves. But after 3 hours, you have an inflammatory situation where you're getting a lot of ATP, a lot of inflammatory cytokines, and an acidification of the airways. And so 3 hours seemed to us the minimum duration needed. So we looked at, well, what if you just delivered normal hypertonic salt, not alkaline, talk about alkalinity in a second. And what we predicted and later confirmed, as I'll show, would happen is that the mucus just lifts off the cilia. It doesn't deaggregate. It doesn't release much globular protein, and so you don't get a very long hydration. We looked also at divalent salts. My work over the last 15 years has shown that to get long hydration with salts, you need divalent cations like magnesium and calcium chloride because they leave the airways by a paracellular route. But even that, you're not getting the long enough hydration. However, if you go to high pH, and given the fact that the mucin are held together in the hydrogel largely by cysteine crosslinks, and the pKa of cysteine is about 8.3. And so some work has been done years ago in pig gastric mucus showing that if you get about 8.5, the mucus dissolves. And so we looked at, well, what if you put in high enough pH to let go of the globular protein? It will reform as the pH gets neutralized, but if you have some period of time where you have a lot of globular protein, and we predicted, well, you should get a lot of hydration. And so we then studied this in vitro, and indeed, this kind of crazy result where you see the top red line, solid line. This is magnesium chloride, pH 9. You're putting about 20% of the surface liquid volume, and you get this really long hydration. Go down to pH 8, exact same composition, and you get much less. So there's a lot more osmolyte, even though you're not putting in more osmolyte, there's a lot more osmolyte in the high pH case. And this and other indicators suggest that you're controlling the release of globular protein, and that's the secret. And we did studies to show that the stress release is 4 to 6 hours, depending upon how dehydrated the airways are. So the last point I want to make here before getting into our clinical data is that you might say, well, is it smart or natural or safe, actually, to be raising and lowering the pH in the airways? And so little known, but published a few years ago, is that in normal airways, when you breathe, there's so much CO2 that's being exhaled that passes through your airway surface liquid on exhalation. There happens to be carbonic anhydrase 12, which is tethered to the apical epithelial membrane. And so you get this big production of bicarbonate. And so, in fact, the pH in the upper airways is fluctuating between 7 and 9 with every breath. It goes away with cystic fibrosis, and it's antimicrobial. And so we're, in some of the work we're doing now with Sangeeta and other colleagues around the world, we're beginning to look at why that is, and it gets to this mucin interactin. But just to say that there is this natural phenomenon that we seem to be recapitulating here. So we then full disclosure, I founded a company with a student of mine, and I'm actually the CEO of the company at the moment. And so my conflict here. So we are developing a new treatment for refractory chronic cough, and we've done two clinical trials, and are starting a third. And it's very simple. It's non-pharmacological, and so we're using the ions that are in the airways. It involves about 20 seconds of breathing, and the treatment is 4 times a day, but we think we'll go down to twice or once a day here, as I'll explain. So I'm just going to quickly walk through the data. We just published the REACH trial data a few weeks ago. This is my first presentation of those data, and then I will try and finish this in 10 minutes, and I'll just do a little closer here. So in our first study, there's a lot to be said. It's really a fascinating frontier cough. This is showing 12 people and their daytime and nighttime cough. And so people do cough at night, which is kind of a drag, and it varies a lot. You get some people coughing a lot and some people cough a little. So heavy cough is 20 or 25 times an hour or more every day of your life. So if you imagine, but some people cough a lot more than that, believe it or not. Most people are around 5 to 10. So what we found is in treating these patients, people got the pH 8 aerosol didn't get much treatment effect. People got the pH 9 aerosol got a lot of treatment effect, near the best treatment effect that's been seen in pharmacologically active drugs. So that was indicating the mechanism of action, as I explained. We started on a phase 2A study I'm going to go quickly here, but it was a double-blinded, randomized, placebo-controlled crossover study. Now, in crossover studies, as you may know, you're counting on the fact that people can get both placebo and active, and there's enough washout between the two that there's no carryover. We had avoided that in our first study because we knew that, well, we're probably going to get some downregulation of mechanosensitive ion channels, and that might take a while, but we didn't know how long. But what we found in this study is it actually takes a long time. And so we stopped the study halfway through, just above the minimum number we needed for the power analysis. And the results I will show here. So we found that the cough rate goes down immediately from the first day, similar to our last study, and just continues to go down with treatment. The efficacy of treatment relative to placebo, we had a big placebo effect for reasons I can describe. But this is near the high mark of efficacy seen in pharmacologically active drugs. Cough bout, you cough more than once every 2 seconds. Turns out that has a big impact on quality of life, and so that we have a much bigger effect on. Do you feel well? It's a big thing for cough right now. The only way that a doctor knows whether you have cough is you come and say, I have cough. And he says, okay, on a scale of 1 to 40, how bad is it? And if it's over 40, then you're in need of treatment. And so now we're only now getting to the point we can actually measure cough rate. This is a big thing. And so to get a 40, this never been seen in a cough study before, to get that big of a quality of life improvement. And we think it has to do with the fact that there's just no drug-related side effects. But this is the kind of the main surprising result of the study that has led to this next study. So you see this cougher who starts out at 25 coughs an hour. The blue zone is 2 weeks of treatment, and you see the cough going down, down, down. This is the daily cough. And then stops treatment and just keeps falling. And so 3 weeks after treatment, there's no cough. So it's like 1 cough an hour is the limit of detection of this digital cough monitor. And so then it slowly starts to come back. And so he gets into the next treatment and it's just roaring back, basically. And so what that led us to believe is that there's an opportunity here to lower frequency of dosing and go to a management of cough. And that's what we're exploring in our next trial. This is just quickly a comparison with other studies here. So what I want to do here in the last 8 to 10 minutes is take a step back. So we're very excited about this new treatment modality for chronic cough. As a scientist and as a human being and a colleague of my friends here, also really concerned about just the future of breathing, right? And so this is not just a matter of cough, it's a matter of actually, and it's unfair, right? And so it's not like we're all kind of facing the same challenges. Where you live and how you live has a big impact on the stress here. So the question is, well, what do you do and how do you understand it? And so I'm just going to, like, 4 slides here, but I'll take a little bit of time just to say that this actually is a really interesting graph. This is going back in time, last 40 years. And we looked with Dan Lee, climate scientist at BU. So we looked at cities in the United States, or urban areas in the United States, and we kind of asked how many days per city were you experiencing a heat wave. And so that number, particularly if you look at the number of days over 100 days, the number of cities getting those numbers of days has gone up like over 100-fold since the 1990s. So it was just kind of drop in the 1990s. But the reason is that there was this volcano that erupted in Australia, which actually brought the temperatures down. So if anybody knows anything about asthma, you will know that the 1980s were viewed as this particularly bad period. Nobody quite knew why. Well, what happened actually was like the air, actually temperatures went down. Everything kind of came roaring back in the 2000s, but there was this kind of anomaly. And just to give a sense of how bad is a heat wave, in the 30% relative humidity case, we were exposing airways to like 4 kilopascals. The exposure in a heat wave is like 6 kilopascals. So these are really very high stress situations. So the looked at another way. So we did also in this last paper a study showing that by the end of the century, independent of heat waves, the temperature will go up to such a degree that 40 to 50% of the country will be experiencing inflammatory or highly inflammatory conditions. And I kind of point out this analogy between the crop failure rate and the asthma and chronic cough incidence. Whatever the growth in prevalence of chronic respiratory disease is probably going to go up multiple fold because of these conditions. So what do you do? So, well, and just to connect my talk, second to the last slide here, I've talked about this kind of mucosal collapse and then highly inflammatory situation. And that corresponds to this sort of, waking up in America with the yellow zone or the red zone, just normal tidal breathing. You're facing these kinds of conditions. Now, what we are discussing right now with Sangeeta, many of you know probably that she's developed a really fascinating diagnostic for a breathalyzer diagnostic for depositing nanoparticles in the airways and sensitizing these particles to enzymes that are emitted in inflammatory conditions. And so what we've been interested in exploring is whether we can take an individual and after an exposure to a heat wave, 8 hours of heat, can we detect inflammation and can we stratify that inflammation and have a treatment that would be rapid? So you go through a heat wave, or your kids go through a heat wave, or you're exposed to wildfire, and you can diagnose that quickly and treat it in a short-term treatment and stop the inflammatory cascade. And as Sangeeta talks about the interception in the cancer context, but intercepting the inflammation is really what we need to be doing. And so we're interested and look forward to the collaboration. So that's it. Thank you very much.